All right, so you can follow along with me on your screen, but it's the same thing. So the biggest lesson this year is the central dogma of biology. It's called DNA to RNA to protein. That three-step process is so important, and you're going to be seeing this on the EOC a large amount, lots and lots and lots. All right, so the DNA to RNA to protein is one of the most important things that you're going to hear this year. You should really pay attention to this section. It's going to be heavily tested on the EOC. It's considered one of their high-ranking test things. They say that they rank them high, moderate, and low, and this one is a high. It's a big deal. And the reason why I have a builder on here is because it's kind of like building a building, if that makes sense. It's how your cell builds things and what the DNA is actually for. All right, so the central dogma itself is just DNA codes and is replicated by itself, but whenever it is translated, it goes to RNA. That RNA is transcribed into proteins. These proteins make you. Your cells run off of proteins. They're building blocks of your cells. It's what makes a cell. A cell is made of protein. You've probably heard of proteins before, especially if you're like a football guy and you drink those nasty, disgusting protein powder shake things, right? Or you eat those bars, the protein bars. Okay, proteins are cellular breakdown components. Cellular components make up proteins, and some of these proteins your body can't make as some of these amino acids, and you need to get those from outside sources. That's where those protein shakes and things come from. All right, so RNA is a little different than DNA, and there's 5 differences between the two, and you need to know all 5. All right, so sugar in DNA is deoxyribose. In RNA, it's just ribose. So what's missing is the deoxy, right? And D means without, oxy means oxygen. So the ribose has the oxygen in it, and the DNA does not. All right, so DNA is deoxyribose, nucleic acid. So it is without oxygen. And then the ribose includes it. It is a special type of sugar. So we're talking about the sugar rings, those sugar phosphate backbones. So the backbone is different slightly. RNA is only single strands. It's not double-stranded like DNA. DNA is that double-stranded double helix. This is a single strand, and it contains a special nitrogenous group that the DNA does not have. DNA has thymine, and thymine is replaced with uracil in RNA. So anywhere there'd be a T, it would be a U in RNA, which is a big difference. And on the test, they will ask you this. Is this a molecule of RNA or is this a molecule of DNA? And the best thing that you can do, look for the uracil. If there's a U in it, it's RNA. That's the easiest way to figure that out. RNA is disposable. RNA is a copy of the blueprint, if you will. All right, DNA is kept safe and housed in the nucleus. You don't want anything to happen to it because that DNA codes for every protein that the cell can make. Therefore, everything that the cell needs, it has to get from the genes, and those genes are on the DNA, right? The number of A to T, G to C, that's super important because that's what gives us that genetic diversity that we learned about last 9 weeks, or not, 9 weeks. All right, so RNA is disposable. You can get rid of it. After RNA is done, it is destroyed. It is done. It's just like a copy. You can throw it away. You don't want to throw away the DNA. All right, RNA can be outside the nucleus. DNA cannot. And again, that's because DNA has to be safeguarded. It needs to be super safe. All right, it has to be protected. So finally, if you're going from DNA to RNA, what base pairs with adenine? Uracil, yeah. So A to U, G to C in RNA. So the thymine is gone. It is turned into uracil. So if I receive this DNA copy, what would the RNA readout be? C goes to what? G. A goes to U. G to C. T to, T to, and then A to. Very good. So you're going to wind up with this right here, GUCAAU. So your RNA is complementary, but has some differences, right? It has a different backbone, the different sugar. It has a different nitrogenous base than the other one. But the RNA is what actually does stuff, okay? You don't actually do anything with the DNA except replicate it or make RNA off of it. That's it. The DNA has to be held safe because it doesn't want any mutations or anything to come along. So the RNA is what actually does the work, guys. All right, so there's a couple of different types of RNA. You have messenger RNA. A messenger is somebody who takes a message somewhere, right? That's basically why we named it that, because it takes the DNA's message for a gene and it takes it outside the nucleus to do something with it. It is a messenger. It takes it. It travels. All right, so it carries the copies outside of the nucleus into the cytoplasm because the DNA cannot leave that nucleus. It's super important to stay safe. Then you have ribosomal RNA or rRNA. And so the ribosomal RNA, take a wild guess what it implies. It means that it has to interact with the ribosome, right? Ribosomes make proteins. We talked about that when we were doing the cells. Everything that we've done up till now has been little steps leading up to this big overall idea, okay? The parts of the cell we had to learn, the genetics we had to learn, and now we're going to learn how all of it worked together to make something, and what it's making is proteins. All right, so the rRNA makes up the ribosome, and it actually is the part that is assembling the protein. Transfer RNA, the tRNA, brings the amino acid to the chain. And you're going to learn that the proteins are just sections of amino acids just stuck together constantly. So it brings that amino acid to the ribosome so the ribosome can put them together. All right, so when you're looking at the big picture, so inside of the cell, so this is giving us an idea of what that cell looks like, kind of, sort of. So the nucleus would be in this section here, this one right here. This would be representing the nucleus, that inner bubble. And so that inner bubble there is the nucleus. And what's happening in the nucleus is your DNA is getting transcribed. These words are very similar, and you have to be careful. What I remember is scribing means writing down. And so it helps me to remember that the DNA is being written down as RNA. All right, so the transcription is when we're making RNA copies of a gene. We don't do the entire DNA molecule. It's 10,000 base pairs long. We don't do the entire DNA molecule. We only do the section of gene that it needs at that point, whatever it's making. So it just is going to transcribe just the section it needs, just that gene that it needs to code for that protein. That's all. All right, so it's just going to make this little bubble. And on that bubble, it only has to unzip the DNA for just a small section, copy that. It'll have a start codon and an end codon, so it knows how far to unzip it, and it's going to transcribe just that little bit. And then now we have a free-floating mRNA. Once the mRNA leaves the nucleus, it gets translated. That's where all the action happens. That's where the rRNA, the ribosome units, and the tRNA, where they're actually assembling and making it. And it happens in the cytoplasm. All right, so transcription is in the nucleus. That's important. That's another tested question that they often ask is, where is transcription taking place? Transcription takes place in the nucleus. Translation takes place in the cytoplasm. Super important that you know that. Transcription just copies the DNA into RNA. That's all it does. So translation is outside that nucleus, and it happens with a ribosome. That ribosome is the rRNA. They are the same thing. All right, and it attaches amino acids to make the protein. So it looks really complicated, but it's not. We're going to break it down nice and simple for you so you'll understand what's going on. Yes, you will need to know the stages of each. You will need to know how they work, especially the translation. So it happens just like a construction. DNA would be your master plan. mRNA is just like the everyday copy of the blueprint that you give to each of the builder people. You know, you get one copy for the plumber, one copy for the guy who's framing it, one copy for the roofer, right? You don't want to give out the master one because there's only one. So you just copy that. And so that's the mRNA. The rRNA would be the actual builder guy because he's actually putting all the parts together. The tRNA would be like the gopher. You know what I mean when I say gopher? That's the person that goes for that. He's the do boy. Hey, go give me the hammer. Go for this, go for that, go for this, go for that. You know what I mean? He's just bringing things to the guy who's actually building. And then the amino acids are the actual wood. So if I'm building it, I'm going to need a bunch of pieces of wood, and the builder's going to put them together, right? So the amino acids are the pieces of woods, and then eventually you will make a protein, which is in this case the building, right? So it's just like building something, just like constructing a building. All right, so this is a molecule of mRNA, messenger RNA. mRNA is transcribed in the nucleus. So script is writing, so I'm copying in the nucleus, all right? It is complementary, but not the same as DNA. When I say complementary, what I mean is that you are able to match up what the DNA used to be based off of the mRNA strand, because A to U, G to C, right? So once it's transcribed, the mRNA can leave those nuclear pores. Every 3 letters is a codon. All right, so every 3 letters is a codon. Each codon transcribes for a amino acid. So translation of the genetic code is protein synthesis. So the first step, ribosome attaches to the mRNA. This is the first step of transcription or translation. What are we talking about? Do we already have an mRNA? Come on. Do we have an mRNA here? Yes or no?
Yes.
Yes. So is it transcription where we're making mRNA? No. It's translation, right? So the first step, ribosome attaches to that mRNA. You need to write that down. The ribosome, if you notice, has got 2 circles on it. That's because the 2 circles are actually in 2 subunits, and one attaches to the top, and it really is bigger than the other one, just like it's shown here, and one is smaller. So they kind of sandwich the mRNA in the middle, and it really does look like that. Step 2, tRNA arrives. And they are called tRNA because they're transfer tRNA, but they actually have this little T-like structure, so they kind of look T-ish. And if you notice at the top, this guy up here is going to be an amino acid. So what it does is it brings a specific amino acid to the first 3 bases. So this is your amino acid up here, this little box of this greeny blue box at the top. And it's going to bring the matching code, brings a specific amino acid to it. And they match that codon. The tRNA, the letters on the tRNA are called anticodons because it's the opposite of the mRNA, right? It's matching, but different. So we call it the anticodon. So the codon is in the mRNA. The anti that matches it is on the tRNA. And that's what attaches them so they can bring the correct amino acid. So that's the next step. The 3 unpaired bases, the anticodon, link up with that codon. They actually stick together. All right, next step. What happens? All right, so another one comes down the chain, brings that second amino acid. It attaches with the anticodon. It links up. And so you wind up here with both of them linking. Oops, hit the wrong button. When they're both next to each other, a peptide bond forms. That's a special type of bond that holds these amino acids together. You do need to know that. Up till now, we've only talked about hydrogen bonding. Hydrogen bonding is not a true bond. A peptide bond is. It is a true bond link. And that peptide bond is very strong. It literally links them and holds them together extremely tightly. So it's important to know that there's 2 types of bonds that they're going to test you on. The hydrogen bonds that hold the DNA together, but then these peptide bonds which hold the amino acids to one another. And then finally, that first tRNA falls off and it scoots down the ribosome. And you get another tRNA is going to come in, and it's just going to keep making this. So the ribosome moves along, and then you get another tRNA comes into place, and then it bonds, and then a peptide bond is formed, and then the first one falls off, and it keeps going and keeps going and keeps going until you wind up with this long polypeptide chain. And it keeps doing this until it reaches what's coded for a stop. So in the code itself, it literally, one of them will code to start. That tells it where it starts going at. And then one of them will code to stop. And that tells it where to stop at. And once it stops, it all breaks off. And then you have a protein that's made. So the polypeptide is another word for protein. Poly means many. Peptide, those are those bonds, right? Polypeptide. It's just a fancier word for protein. Same thing. So once the polypeptide is complete, it falls off and it will be translated. So it's not that hard to figure out how it does it. The tRNA brings it in there and it works this way. But the questions on the test are going to be more related to like, can you figure out using these tables, what is the polypeptides being translated in this specific mRNA? So you're going to have to use one of these 2. I have seen both of these on the test. So you have to be able to use both of them. All right, so on this one, when you're using it, I went too far. All right, so your first letter is here. Second letter is the medium size, and the itty bitty ones around the outside, those are your third one. And then the names of the 20 amino acids. So your body makes, or any of the cells, make 20 amino acids. And these 20 amino acids are enough to where you can change them out in an almost infinite way so that you can make any protein that your body needs. All right, so the first one would be in the middle. The second one would be on the next ring. And then the third one would be the outermost smallest ring. You get how that works? And you just match them up, whatever it is. So let's say that we're going to do GAU. Oop, I can't write. Well, G, so first one's a G, then to the A, then to the U. It codes for aspartic acid. Do you see how that worked? All right, so if over here on this one, see GAU aspartic acid. So on the second one over here, this one, I've also seen them use this one. So let's do UAG. All right, so UAG. So the first letter is on the left-hand side. So you go to the U. Second letter is on the top, so I go across to the A. And third letter is on the bottom, so I go down to G. UAG is a stop codon. Do you see that? So what is a start codon? What's the start one? Look at your screen. Which one is the start codon? Oh, it doesn't show you on here, does it? Shows you the stops, but it doesn't show you the start. All right, but there's 3 stops: UAA, UAG, and UGA. Those are the 3 stop codons, and that triggers the cutoff of the tRNA, and it stops the polypeptide production or the protein production. All right, so let's use these. Try this one. What amino acids would be coded by the following DNA sequence? So let's use this one over here. So where should I go first? CGG, right? So I go to the center and I go to C. Then I go out a ring to what's the next letter? G. So it's there. And then G again, it's arginine. See it? So the first one is arginine. What's the second one? What's the second one? So the second codon would be ACC. So what's the second one? Say it out loud.
Oh, nice.
Spell it then.
THREONI.
Yeah. All right. Threonine. Threonine. All right. So the way he did that is he went A to C is threonine. Threonine. All right. What's next? GCT. Ah, so what are you noticing we screwed up with then? Oh. What did we do wrong? What's the issue? This says DNA sequence, doesn't it? So I wanted you to do that. Because if it's DNA, what do I got to do first? I got to convert it to mRNA, don't I? Kind of wanted you to run into that wall on purpose. So because this is DNA, you have to convert it to mRNA and then use the codon chart, okay? So I gotta unclick this. Hang on. Unclick this. Will you let me do this? Maybe? No? There we go. All right, because it says DNA sequence, so we need to make this an mRNA strand first, right? So what does C turn into? Come on. A turns into what? Which not an RNA, it turns into U, And then G turns into C, right? So C goes back to G, G goes to C, A goes to U, C goes to G, G, C, G. What is this going to be? It's A, U, A, and then G, right? So you're going to wind up with a mRNA sequence of this. All right, so now on your paper, I want you to use, like we did, and actually figure out the correct amino acids. Remember, each 3 is a different one because that's a codon is 3.
Move back there so I can see.
Yeah, baby, you certainly can. You also have it on your screen if you need to. All right, so on the first one, you're going to do GCC, then UGG, then CGA, then UAG. So what did you get for the first one? What's the first amino acid? Say it again. GCC. It's okay if you don't say them right. They're crazy names. This one?
Yeah.
Right? So I'm just going to go ala, because it's alanine, right? I don't have enough room to write all of it. So you're going to have alanine. Because GCC makes alanine, right? All right, what's UGG? So you start U, G, G, tryptophan. So trip. All right. What's CGA?
Let's see. What'd you say? What?
This codon here. CGA.
Hold on.
What? Nope.
CG.
CG. C, G. What's the next? A. What is it? Arginine. Arg. I'll just do arg because I can't write it all. All right, and then what's UAG? UAG. Stop. Right? And that makes sense because a protein does what at the end? It's a stop, right? So this one would be UAG. That's how they got stop, right? You see it? It's not that hard. You just have to have one of these charts. They will always provide you with a chart. Now, I can't tell you which chart they'll give you because they could give you this one, which I think is a little harder. That's why I chose it to show you, than the other one, which is pretty simple. All right, so we're going to know what would be the mRNA. Let's go backwards because they are going to ask you both ways. What would be the mRNA sequence for a polypeptide chain that has the following amino acids? All right, so if you're going backwards, lysine, cysteine, proline, you would have to go backwards, wouldn't you? So you would find lysine. So I have lysine here. right? See it? So I could get lysine. My first letter would always be what? A. Second letter would always be A. What is the third letter, though? It would be G or A, wouldn't it? So there's two options for G or A. But is this the DNA sequence like it asked you? What sequence are we coding here? What sequence would that be coded? So we know that it has to be an A, and a G or A. But what is this sequence? Is this DNA or mRNA? mRNA, right? That is the mRNA. So what would the DNA be? Go backwards. So if this makes RNA, if I'm going back, it goes there, right? So if this is an A, what would it be in DNA? T. So it would be TT and either C or T, right? One or the other. And you just keep doing that. So this bottom one here would be DNA. So this is, the top one would be the mRNA strand, and then you go backwards to make the DNA strand, which in this case would be TT and either C or T, right? You got to be able to go both ways. And then you can figure out for the other two. It's not that hard. You just do the same thing. And you got to do the mRNA before you go to the DNA. All right, so if you need to do the other one, let's try one with this one over here, though. Let's see, what if I do using this one here, so not this one. We're going to use this one just to get you used to it. Somebody give me a sequence. What do you want to do? So give me a DNA sequence. What you going to get? Give me one. All right, so you want to do TAC. All right, what else? G. She's just giving me random letters. Come on, what else you got? TA. CG. What do you want the last one to be? What? How about just one last one? So we'll do A. There we go. So we've got so the DNA strand is going to be TACGTA, TACGTA, CGA. All right, so that's our DNA strand. All right, what is the mRNA strand that corresponds to this? So what's the mRNA then gonna be? T goes to what? Uh-huh. C goes to? There you go. So you're going to wind up with AUG, CAU, GCU, right? And then what are the amino acid sequence then? So I'm just going to shorten this A, because I'm not going to write out amino acid sequence because that'd be ridiculous. So what is AUG? Now use this one over here. Use this one to see, make sure you can do it, all right? So what is AUG? So first letter A, second letter U, and then G. So G tells me it's this one, isn't it?
Yeah.
Methionine, met. That's methionine. You see it? All right, next letters. What do we got? What's our next codon? CAU. All right, so C. So you're going to go C. A is the second. And then what was the other one? U. CAU is his, right? All right, so histine. Or histidine, whatever. And then what's GCU? So you go down to G at the bottom, over to C, and then U. What is that? Alanine. See it? So she went G, right? C, and then U, GCU. See it? For alanine. So I've seen them use both codon charts. So you have to be able to use both. All right? Doesn't matter which one they give you, you should be able to use it. As long as you remember that the first letter starts in the middle on the circle one, first letter starts on the left on the chart version of them. All right? And this is only for mRNA. So if they give you DNA, you have to convert it first. If they give you RNA, then you can use it straight on. So be careful. Don't make the same mistake that we did on the first one, okay? I did that on purpose because I don't want you to screw it up. It depends on what they give you. Did they give you DNA or did they give you RNA? It's not so bad, is it? This is literally how life is formed. All right.